In this chapter, we dive into the building blocks from which life is formed, and we also talk about some of the characteristics of living things. To start, what are the things that make something alive? There's a lot of different attributes here, but I've included three of some of the bigger ones. The first is that living things are able to maintain what's known as homeostasis. This word basically just means unchanged, standing. Now, to illustrate this principle, consider this. This is a picture of a very cluttered garage. This isn't my garage, but it very well could be my garage. For whatever reason, my space has become very cluttered. And contrast that to the spaces that are governed by my wife. They stay very nice and neat and orderly. And as soon as something in her space becomes cluttered, it's immediately brought back to being nice. As soon as something in my space becomes neat, within a moment's notice is turned back to clutter again. And so we have these two different environments, those governed by me, which are cluttered, and those governed by her, which are not cluttered. In either case, they stay that way generally. Okay, so that's a little bit about what we mean by homeostasis, is that inside of a cell, for example, the conditions are much different than the conditions surrounding it. And despite the flux and the change in the conditions surrounding it, it's able to maintain its own environment, much like how Zach can maintain his cluttered garage, even though his home is very nice and orderly. Okay, now the way that this is accomplished is a little bit clever. Consider this. This is a picture of I believe, a paramecium. Everything inside here is considered living. Everything outside of it is considered not living, so frowny face there. And what is it that separates these two? Because this is mostly all just water with stuff in it. This is mostly just water with stuff in it. And so what is it that makes the stuff in water here different than the stuff in water out here? The answer is that there's a boundary. There's a partition. Okay? And this is this boundary, in its simplest form, in the simplest, single cellular living organisms, the boundary is created by what's known as the plasma membrane. And the plasma membrane is not a water type loving substance. It's actually made principally of an oily material. And so, all of us know if we put a drop of oil in water, the oil and water don't mix. So this little film of oil that wraps around the cell allows the stuff in here to not mix with the stuff out here. And so thereby it can separate its environment, and not only separate it, homeostasis doesn't just mean to have separate environments. Homeostasis also means to maintain the environment. So whatever conditions are inside this cell, it can maintain them. So the right pH, the right amount of saltiness, the right, oftentimes in more advanced organisms, temperature, all the different conditions that make the operations of the cell optimized, those conditions can be maintained. That's what we call homeostasis. Now, the partition of the cell and the thing that keeps it isolated from its environment so it can maintain homeostasis can vary. Some of these plasma membranes can be very extensive. For example, this environment here appears to be very hostile to a cell. It looks very rigid and rocky. It almost looks like an exogorth from The Empire Strikes Back is going to wiggle its way out of this cavern here. What are we looking at here? This is, I'll give you a moment to see if you can guess it. This is actually, I'll give you a hint, it's in your body somewhere. This is a picture of an osteon, a scanning electron micrograph of a osteon, which is the functional unit that makes up your bone. So all of your bones, the compact bone at least, is going to be formed from these structures called osteons, which basically look like these concentric rings of bone that span all the way along the length of the bone. In the middle, there's a Haversian canal, which basically is a place that blood vessels can pass into and out. And then because your bones are so dense, it's hard for cells to live in there, but there's these little pockets or these caves that are created in the bone material in which the cells can live. But one of the caveats is that while there might be blood vessels running up and down this empty space, there's still this dense wall of bone between the source of nutrients, which would be the blood vessel, and the cell that's living in here. So if you'll note, if you look really close, you'll see it looks like there's all these little tunnels. Those are called canaliculi. And when we look at the cell, the osteocyte that would actually be living in these caves, it's developed a very elaborate plasma membrane to ensure its homeostasis. Look at all of these sprawling branches of the cell membrane coming off of the cell, which is right here. Those branches, they find their way down in through these caves, and they make their way to the Haversian canal, which is where the source of nutrients come. So it's like they've got all these like little pipelines that pipe the nutrients right into the cell. So can be very elaborate structure there, the plasma membrane, and the way that the cell is going to stretch out its far-reaching processes to get to the source of nutrients and to maintain its homeostasis. Another characteristic of living things is that they can reproduce. So this brings up or beckons a kind of a discussion here of viruses. So here we have a picture of a coronavirus. Note the little spike proteins, which make it look kind of like a crown. That's where the name comes from. Corona means crown, I believe. And viruses are tricky little buggers because they actually can't reproduce on their own. For example, if you had a vat of virins in a tube and you left them there, they would never reproduce. They have to infect a host cell in order to hijack their cellular machinery and then use it to reproduce. So viruses in and of themselves are not considered living things. Only are they alive when they infect a host. So there's an interesting distinction there between what's a living thing and what's a not a living thing, but all living things should technically be able to reproduce. Okay, all living things can respond to their external stimuli. This can look different in a variety of ways. A couple of examples here would be something even as simple as this, which is a protist, a single cellular protist named a euglena. And euglena have a little eye spot. This tiny single cellular thing has just this small little area that can detect light, which I just think is totally fascinating. And using that eye spot, it can find its way around the universe. It usually can go either towards greater concentrations of light or further away from concentrations of light. And that concentration gradient of light can be detected by that single little eye spot and used to determine how it moves around in the world. So isn't that cool? Just one single little cell right there. One single little cell. A big cell, but a single cell nonetheless able to find its way and respond to external stimuli. Okay, another great example of this is sperm cells. Sperm are amazing features of ingenuity. Okay, amazing feats of ingenuity. They are able to find very, very, very specific spot in the human body and do so in an extraordinary amount of time. So these sperm cells, it's kind of interesting. When they are made, they're just basically inactive zombies. They don't really move at all. Okay, and then it's right prior to ejaculation, they come in contact, at least in humans, they come in contact with seminal fluid, which turns them on. They become swimming machines. They swim like crazy, and they have an incredible feat because should they need to fertilize an egg, they have to traverse through the uterus and all the way up the fallopian tube, and they have to pinpoint a tiny, teeny little microscopic cell. And how does a cell without an eyes or ears or like sonar echolocation, how does a cell do that? So just to give you a reference point here, the path of a sperm to an egg, the total path, if we consider the whole length of the uterus and the fallopian tube, it's about 20 centimeters. Okay, that's 200,000 micrometers. A sperm cell is 50 micrometers long. So if you do the math, 200,000 divided by 50, that means that the path that a sperm has to travel to fertilize an egg is 4,000 times greater than the length of the sperm. So if we were to apply that to me, I'm about 6 feet tall, so 4,000 times 6 is about 24,000, which ends up being about 4 to 5 miles. Okay, so a sperm fertilizing an egg is like the equivalent of me walking 4 to 5 miles. A sperm can make this journey in about 30 minutes. The fastest swimming sperm can make that journey in 30 minutes. I would I would never be able to go 5 miles in 30 minutes. Okay, I could maybe do 2 miles or 3 miles in 30 minutes, but I don't know if I could do 5 miles in 30 minutes. I'd be running pretty hard. Maybe if my life depended on it, I could run. I mean, many people can make 5 miles in 30 minutes, and I mean, what is that? That's like a 7-minute mile, or like something like that, a 6-minute mile average. But even consider this: sperm are doing this swimming. Okay, I could never swim 5 miles in 30 minutes, never. And not only this, but they're also swimming basically uphill. It's like an infinity pool that they're going the opposite direction in a way. So, and then consider this: like, let's say that even I was a really great swimmer and I could somehow swim 5 miles in 30 minutes. Let's imagine that the 4 to 5 miles I have to swim is on a really big lake, and I have to somehow touch a buoy that's 4 or 5 miles out in the lake, and I have to keep my eyes closed the entire time and somehow find it. Okay, I could never do that. I could never swim 4 or 5 miles and touch a buoy, with my eyes closed the entire way. But sperm can do this. They can pinpoint the exact location of the egg because of the fact that the egg releases different compounds that they can detect, and they can respond to it and adjust their path of travel to accommodate for that external stimulation. Okay, so those are some of the properties of living things. Now we're going to get into the actual framework, the composition of living things. And I like to think about this class as kind of an escalator, where at the very top floor, we talk about communities of living things. Communities are made up of populations. Populations are made up of individual organisms. Organisms are made up of individual organ systems. And so if we ride our way all the way down the elevator, we're starting kind of in this class at floor number 1. And eventually we'll work our way up to the top of the building. But for now, we're starting at the ground level. We're going to be looking at the very thing that makes up all of the stuff from which you are composed, or at least from which your body is composed, and that would be atoms. So we'll talk about a little bit about atoms, and then we'll talk about the different types of molecules that atoms can make, which would be found in your cell. All right, atoms, there's a lot of different kinds of atoms, and this is always something that people find a little bit confusing. There is an atom, there's an atom, and that's not necessarily the same thing as an element. These words are similar, but not exactly the same. So elements, the way that you could think about those they're the different types of atoms. So an atom is the smallest building block from which matter is made, but there are different kinds of atoms. Let's think about it like Lego blocks, right? There's a lot of Lego blocks are the building block, but there's a lot of different colors of building blocks. There's different types of the building blocks, and that's similar with the atoms. Now, the different types of atoms that exist in the world are shown here on our periodic table. So you can see we've got close to 120 elements shown on here. So there's about 120 different kinds of atoms that arrange themselves in different combinations to make up all that we see in the world. The specific types of atoms that make you up, I mean, there would be many of these on the table, but by and large, a majority of them are actually just a few of these. 62% of all the atoms in your body are hydrogen. 23% of all of your atoms in your body are oxygen. So between, that's pretty much hydrogen and oxygen make up the majority of, your body. And that makes sense because a lot of what you are is water, and water is hydrogen and oxygen. Okay, a lot of you is made up of carbon, 12%, and then a smaller percentage, but still a very large percent, is 1.1% nitrogen. 0.2% is made up of calcium, and then phosphorus makes up the other percentage, and then sodium, and then also sulfur. Okay, so carbon, nitrogen, oxygen, hydrogen, sodium, calcium, phosphorus, and sulfur make up a majority of your frame. There's a whole bunch of other ones, but we won't go into those because those are more the micro dose. They're the micro composition, smaller percentiles. All right, you don't have to know any of these percentages. I just put it up there because it's a little interesting. All right, here's an example of an atom. This is an atom of, well, I won't tell you what it is an atom of. I'll see if you can figure that out later. But in either case, this is called a Bohr model because it shows a couple of different things. It shows the inner nucleus of the atom, and it also shows the electrons that are whirling around. So all atoms have a few different parts to them. There's the protons. The protons would be positively charged, and they would be found in this little nucleus area. Okay, so you get, in this example, we have two protons, two little particles. Now, protons, the number of protons, this is kind of a couple of different important features here of protons. They are positively charged, as we said, but another really important thing is that it's the number of protons which determines what type of element it is. So because of that, we call the number of protons an atom has its atomic number. If we go back and look at this periodic table, the atomic number is listed here in the top right of all of these. What that means, this number specifically means, is how many protons are in the nucleus of each atom. So if you look at an atom and you find that it has 17 protons in it, then that atom, the specific type of atom that it is chlorine. And as soon as it gains or loses a proton, then it shifts and it becomes a different atom. And atoms can change if you if you pop a proton off or put a proton back on. It can change what type of atom it is. Okay, so that's protons. Now, another thing that they have are neutrons. Neutrons are a neutral charge. And they are also found in the nucleus of the atom. Electrons are negatively charged. So if an atom is electrically neutral, it will have the same number of protons, because those are the positive charges, and the same number of electrons. So that's what we see here. We saw two protons, and then we see two electrons. Now the electrons are not in the clump in the middle. They're not part of the nucleus of the atom. They whirl around and whiz around in different orbits around the nucleus. And there's a lot of weird physics concerning electrons. There's some theories in quantum physics that say that electrons simultaneously travel all the paths of the universe at once. So it's like when we start talking about how electrons behave at the nano level or the quantum level, it gets pretty weird. But in the case, for our purposes, there's positive charges, there's neutral charges, and there's negative charges. These are in the nucleus. These are whirling around outside. These, as you can see from the picture, are bigger. So these actually have a bigger mass to them, relatively speaking. We usually give them a mass of what's known as 1 AMU. And electrons are so small, we don't really consider them to have any mass. So if we look back at the periodic table, all atoms will have a atomic weight listed. This basically tells you something about how heavy they are. And how heavy they are should be the number, what determines how heavy they are would be the number of protons plus the number of neutrons. The electrons have a mass so small that we usually consider it negligible. Okay. Now, how atoms connect together is very important, because it's the connecting together of atoms that makes molecules, and molecules and macromolecules make up your cells, and your cells make up you, or at least your body. How the atoms arrange with one another is what we call the nature of their chemical bonds. This is determined by basically the electrons. So whereas the protons will determine what an atom is, how it links up with other atoms is really determined by the electrons. There are a couple different types of ways that atoms could connect. One is called an ionic bond. So consider this. Here we have an atom of sodium, Na. Here we have an atom of chlorine. Now the protons and neutrons were not shown here. We're just seeing a picture that illustrates a nucleus, but we are seeing the number of electrons. So here we have 2 electrons in the first orbit. This is another component too, which we won't really get into in this class, but there are different shells that each atom has, and they can get filled. So the first shell only takes 2 electrons to fill it. The next shell takes 8, so that's why if you count these up, there should be 8. 1, 2, 3, 4, 5, 6, 7, 8. And then generally, an atom is going to strive to either completely fill its orbit, or if it can't fill its orbit, it's going to try and empty it and just get rid of it altogether. So in this case, we have 1 electron here on the outer shell of sodium. This shell also requires 8 electrons to fill. So it's only got 1. So it's going to take a lot of effort to fill this shell with electrons that it's stripped from other atoms. So it's actually easier if it can just get rid of this electron. If it can get rid of it, then this shell goes away, and then its outer shell is full of electrons. This is called the octet rule. Basically, the outermost shell or orbit, the outermost shell of electrons should be full. And in the second shell, it usually takes 8. All right. I'm not going to ask you about those types of questions on the exam. That's a little bit more chemistry, but it is just interesting to note. That is the reason why we kick off an electron. Okay, now if we look at chlorine here, it's got 2 in its first shell, so it's full. It's got 8 in its second shell, so that's also full. The third shell requires 8 as well. So if we count up how many it has 1, 2, 3, 4, 5, 6, 7. So it only has 7 in its shell, and it wants to have 8. So it's very happy to take this electron that sodium has released. So there's a great little mutual partnership that happens here. Sodium wants to become more stable by getting rid of this electron. So then it can just get rid of this whole shell altogether, and then its outermost shell will be this one, which will be full. Chlorine wants to take that electron because then it can stick it right here and have a full shell. So at the end of the day, after this exchange happens, we look something like this. We've gotten rid of this empty shell. Now sodium's got a nice full outer shell, and chlorine has a nice full outer shell of electrons. One of the things that happens, though, is that because we're losing a negative charge here, this was electrically neutral, but since it lost a negative charge, now there's 1 extra positive charge, so it acquires a positive charge. Chlorine was electrically neutral, but now it's going to receive a negative charge, so now it takes on a negative overall charge. So this one's got 1 more electron, this has got 1 less electron. And because now that these have two different charges, they are attracted to one another. Positive and negative charges want to come together. And so it brings these two atoms together. This is called an ionic bond because it's a bond between what we would call our ions. And an ion is an atom with an electrical charge to it. Okay? So when sodium loses its electron and gets a positive charge, it's now called an ion of sodium, a sodium ion. And when chlorine takes that negative charge and develops a negative charge, it becomes also an ion. The two ions bind together. And what they'll do is they'll be doing all sorts of sodium and chlorine, all sorts of electrons from sodium atoms will be given to chlorine atoms, and eventually they start to form this crystalline structure of atoms bonded together. And before long, you have this stuff. Any idea what this stuff is right here? These little crystals are made from chunks of chlorine and sodium atoms, and this is table salt, sodium chloride. So that's an example of an ionic bond. Ionic bonds are some of the weaker bonds, or they have kind of a mid-strength. They're not super weak, but they're not really all that strong either. You know, if we were to put a drop of water in here, very quickly these would dissolve away. Okay, contrast ionic bonds to covalent bonds. Covalent bonds are bonds in which electrons are shared. So ionic bonds, the electrons are given away from one atom to the next. In covalent bonds, the electrons are shared. So here's kind of a nice little video of that. We see an atom here that has one electron. We also see another atom here that has more than one electron. This atom, if it's going to fill out its first shell, it wants to have 2 electrons, not just one. And this one, it's got 1, 2, 3, 4, 5, 6, 7, so it wants to have 8. So occasionally it will dish these over here, and the 2 will whirl around, and then they'll come back over here and whirl around to help make that more full. So when these electrons are shared like this between the two atoms, you know, sometimes being over here and then coming over there, like you can see this is going to come over here and then it's going to dish out 2 of them over there. When their electrons are shared between the two atoms, it's called a covalent bond. Covalent bonds can either be known as polar or nonpolar, and the difference between the two refers to how equally the electrons are shared between the two atoms. So in a polar bond, the electrons are not shared equally between the two. And in a nonpolar bond, the electrons are shared equally. This is going to be super important. Put a bunch of stars all over this, because it's this very simple feature of polar versus nonpolar bonds, which is going to have massive consequences in the way that your cells operate. The biggest thing to note is that polar bonds are hydrophilic, meaning molecules that have polar bonds in them are hydrophilic. This word hydrophilic literally means water loving. So any molecule that's got a polar bond in it is going to be something that likes to be around water, that it can be dissolved by water. Anything that's got a nonpolar bond in it, we would say is hydrophobic, which literally means water fearing. So if there is a molecule that does not have any polar bonds in it, but just has nonpolar bonds, then it's likely not going to want to be around water. Okay? I'll talk a little bit about why that is in a minute, but if you don't understand exactly the science of it, what's most important is that you just know polar bonds, the electrons are shared unequally, they are hydrophilic. Nonpolar bonds, the electrons are shared equally, they're hydrophobic. If you can remember those, all of that, you should be set for the exam. Okay, so let's talk about this for a minute. Like an example of one that would be polar, a molecule that would be polar would be water. And an example of a molecule that would be nonpolar is methane. So let's talk a little bit here about water, and maybe we'll understand a little bit of this distinction between polar versus nonpolar as soon as we're done talking about water. Now, water is a very interesting compound because it's known as the universal solvent. This word solvent is a word that just means any type of fluid that can dissolve stuff, or any kind of fluid into which other things can dissolve. And the stuff that's actually dissolved is called solute. So 2 chemistry words here for you. There are other liquids in the universe, like you could have liquid nitrogen, basically anything in theory, if it has the right conditions, could somehow become a liquid. You could have molten liquid aluminum, and you could have lots of different types of liquids out there, liquid mercury. But water as a liquid is very unique because it's a really, really, really great solvent, and it's not hard for water to maintain its stability. We don't have to like crank on water molecules to make them a liquid that dissolves stuff. Now, the reason why water is so great at dissolving solute, such as the sugar that you would put in your coffee, so in this case, the sugar is the solute getting dissolved in the solvent, the water. Okay, the reason why it's such a great solvent is the fact that it has some pretty basic and powerful polar bonds. So water is H2O, which means that there are 2 atoms of hydrogen, 1 atom of oxygen. So if you look at it, it looks something like this. Here's our molecule of oxygen, or sorry, our atom of oxygen. Here's our 2 atoms of hydrogen. Well, each atom of hydrogen has 1 electron to it, and then an atom of oxygen has 6 electrons. So here they are, 1, 2, 3, 4, 5, 6. And you can note that the electron in hydrogen is going to share, it's going to share its electron with oxygen. This other one is going to share its electron with oxygen. So this forms a nice little happy partnership, because momentarily these 2 electrons can whirl around the hydrogen to make it feel like it has a full orbit of 2 electrons. And then another moment, these 2 could whirl around this atom of hydrogen to make it feel like it has a full shell of electrons. Again, the very first shell is only going to require 2 electrons to fill it. Then oxygen, for a moment, can take all of these electrons and have them whirl around it for a moment, so that for a moment it feels like it has 8 electrons, even though it only has 6, because it's now sharing with hydrogens. It has 8 that it can send across and around its nucleus to make it feel happy. And so everybody's happy for a moment. Okay, so these electrons momentarily pass around. Now, to get to the underpinning of why bonds are called polar versus nonpolar, we turn to this word here, electronegativity. Electronegativity basically just refers to how you could think about it being like how selfish an atom is, how self-centered it is, how eager it is to take those electrons and hoard them all to themselves. So oxygen has a high electronegativity, relatively. And every atom has a pretty well established electronegativity. It's just a number, a score, that basically says how fiercely it holds on to electrons. So what that means is that oxygen ends up holding on to, even though hydrogen has come here to be such a team player and donate its electron, oxygen is going to steal them, and even though they will temporarily whirl around hydrogen to make it happy, oxygen is going to keep them more often than not around its own nucleus. So what it happens is it develops a slight negative charge over here, and we get because of that, we also get a slight positive charge on the hydrogens. Positive charge here. Okay, so even though they're sharing electrons, they spend more, the two negative charges that hydrogen has given up spend more of their time around oxygen than they do over on the other side. So we get a little bit of a negative charge on this side of the molecule, a little bit of a positive charge on this side of the molecule for water. These are called poles. So in this, think about it like in the same way like a battery. A AA battery has a positive charge, a terminal, and a negative terminal, and we call these poles, electrical poles. That's similar for a molecule such as water that has a slight negative charge and a slight positive charge. So we call it polar. This is a polar molecule because it's got some slight charges to it because of the fact that oxygen is one of the atoms that makes up the molecule, and oxygen is a self-centered electron hoarding jerk. Okay, but because of that, it makes for some very interesting chemistry. So because water is polar, it likes other things that are polar. Charged things, charged molecules like to be around other charged molecules, whereas non-charged molecules don't really like to be around charged molecules. So when we think about salt, for example, right here, because of the fact that we have so many positive and negative charged particles in here, and we've got water molecules coming up on the scene surrounding these crystals, and they've got positive poles and negative poles and positive poles and negative poles. These positive and negative charges will strip off, like the negative charge would strip off the sodium, and whereas the chlorine would be tempted by the positive charge. So the charges can kind of work together to strip apart these atoms, which is why a crystal like this so easily dissolves in water. Okay, so a molecule that would be considered nonpolar is methane. Methane looks like this. It's got carbon with 4 hydrogens on it. Carbon is not very electronegative. So in other words, it does not hoard the electrons like oxygen would. So the electrons are going to whirl around the carbon atom just about as same amount of time as they would whirl around the hydrogen atoms. And so because of that, there is no electrical pull on here. There's never a point on the molecule where there's one side of the molecule has a positive charge and the other side of the molecule has a negative charge. So something like this is called nonpolar. And generally that is the case. Anytime we have carbon on the scene binding with hydrogen, nonpolar. Anytime we have oxygen on the scene binding with hydrogen, it's going to add some polarity. All right, so keeping all of this in mind, then we turn our attention to the macromolecules. The macromolecules are the molecules that are going to basically form your cell.